All right, everybody, welcome back to class. It's 8:05. We should get going. Today, what we're going to do is we'll finish up chapter 1. But then what I'd like to do is talk about stress versus strain curves. For instance, we started talking about muscle, but I don't really think it's the end of the story in regards to muscle, because for the most part, muscles are typically going to be attached to a tendon. They even call it the muscle tendinous unit. They could even call it the muscle tendinous system for all that matter. But I think if we're going to have a discussion about muscle, we should also have a discussion about tendon. And if we're going to have a discussion about tendon, we should compare and contrast that with a typical or some other tissue that's somewhat similar, which is ligament. But if I gave you a chunk of tendon, like literally like a dice sized chunk of tendon, I gave you a dice sized chunk of ligament and muscle, there's not too much you could tell me about it. So just looking at or touching the material might not necessarily give you the full understanding of what the material is and how the material is going to break down over time. Now, typically, any material is going to break down over time. It's just how that material breaks down over time is what we're going to talk about today. So let's say, for instance, you has anybody ever like watched a football game or a different sporting event, and they talk about how one athlete tore their ACL with a non-contact injury, like they're just running down the field. Like that's how Wes Welker hurt his knee. And then, every year in football, you'll see like a defensive lineman will sack the quarterback, and then he'll go like 5 feet into the backfield and do like a celebration. And when he lands on the field, he tears his ACL. What would the mechanism for that injury be? Why would that happen? And what are the differences between, say, for instance, when you tear an ACL, but when you injure your bicep, what do they call it? Brianna, what do they call it when you injure your bicep? They call it a bicep tear or a bicep. Usually they'll call it a bicep rupture. So why would they call it an ACL tear versus a bicep rupture? And what happens to muscle when it breaks down and becomes injured? So right now in our discussion of muscle, we're talking about muscle that's 100% healthy with the actin and myosin. What would happen if a muscle was 100% intact and healthy? How does it function? But the flip side of that discussion is what happens if things go wrong. So hopefully we'll be able to use stress strain curves to show you a little bit about each of those materials and what would happen as those materials are loaded with a particular force over a particular period of time. So I think we can use stress strain curves to do that. But then you can also use stress strain curves like Tatiana. I think you're going to be going to grad school for physical therapy. It could be the case that at some point, maybe, you have to work with somebody who has a below-knee amputation, and you're going to have to provide an orthotic or some type of prosthetic or something like that. So maybe you can use the concept of stress strain curves to figure out what materials you might want to use or why you would want to use that. The other thing is, I don't know if anybody experienced this the other day, but talking from 8 to 10:45 was totally exhausting. And I think that it was probably not 100% necessary, right? So what I'm going to do is I'm going to give you a great course. I'm going to do everything I can to make this one of the best courses you've ever had. But when we're done for the day, we're going to be done for the day. All right. I think that sometimes forcing information just because of a time clock makes it so that you guys don't have enough time in your day to do what you need to do. And then all of a sudden there's like a reduction in the validity and the importance of the information. All right. What I do want to do is I'm not going to ask you any questions on an exam unless it is in the book. I'm not going to ask you any questions on the IRAT, GRATs unless it is in the book. Unless we go over it specifically from a book that I give you, because I don't think that's fair. But I also don't think it's fair to give you guys an education solely from a book. So there are experiences that I had. There are things that I learned about. There's reasons why I feel as though like maybe I'm, 44 years old. I'm a little bit older than you guys, but pretty much we're the same person. We're interested in exercise sciences, exercise physiology, the health of the human body. So why is it that I'm still willing to go for 2 hours and 45 minutes and teach about it? It's because it's exciting to me. And the reason it's exciting to me is not because I'm able to follow along in a PowerPoint, but because I had other professors in my life that were just totally amazing. It got me to think other ways and got me to think outside the box. And no person is the same. No person has the same percentage of type 1, type 2 muscle fiber types. Nobody has the same tissues. Everything is going to be different. So to be able to think outside the box and to have the freedom to think outside the box, to me is really what my doctoral program taught me. So what I want to do today is, does anybody have any questions about what we talked about the other day in terms of the different theories of motor control? Right, we have a class right now on exercise physiology. Everybody's going to be studying about exercise. Exercise is movement. So you have to have a discussion on how do you move. Maybe what we should have done is even started the class with some rules of motion, like Newton's laws of motion. I don't necessarily want to get into that, but if we're going to have a class about motion and movement, we should probably hammer down some of those things. So the number one thing is, how do you move? Right? And there are many different theories about how you move. You're going to have to investigate and learn different theories and then buy into one. The theory that we're usually taught in high school is hierarchical motor programming theory. The brain controls everything. But as you do more and more studying, you're going to realize that there are reasons why that theory breaks down. For instance, infinite regress, the degrees of freedom problem. There's other issues like context condition variability, control problems, organization problems. And the more you're exposed to these issues with these theories, the more you're going to say, man, I wonder if this theory is right. And there's other theories out there that you're going to have to be exposed to as well. So I just want to have a little bit of discussion the other day about how is it the case that you can seemingly move instantaneously, or how is it the case that some of your movements seem to just emerge out of nowhere? And I think that the dynamic systems approach to motor control does a better job explaining some of those things. But I would say probably for the rest of at least this chapter, or pretty much for the course, we're going to have to stick to the hierarchical motor programming kind of theory design so that we can go through the course and make sense of the book and things like that. But if you read carefully in the book has little nuances of, like, it says if you look at page 51, it says right on it, the ATP phosphocreatine system, glycolytic system, oxidative system, right? It's use interaction of energy systems. So you have these little hints, you have these words, systems everywhere. What does that mean? Why do they use it? And what is the power of systems? So that's kind of what I tried to get into the other day, maybe get you to think outside the box on the first day of class. But does anybody have any questions about anything we talked about? All right, if not, what I'm going to do is I'm going to share my screen. I'm going to finish up the very best that I can in regards to the PowerPoint presentation. Then we'll get into some stress strain curves, and we'll see how long that takes us, okay? All right, so I'm just going to open up that PowerPoint presentation for chapter 1. All right, and I think that we pretty much got to, hey, chill out. So we pretty much got to, I would say, the beginning of our actin and myosin discussion. So we discussed how muscle has this hierarchical design where you have this bigger to smaller organization. So you have the whole muscle, but then this muscle is separated by those epimysium, paramysium, and endomysium, these little sheaths, these protective sheaths that prevent the muscle from experiencing too much tension, too much force, because too much force, like too much force on a rope, is going to make the rope start to potentially fray. So these little outer sheaths protect the muscle from unnecessary force. It's also where a good amount of the vascularization and the blood flow is. Then the muscle is sectioned off into other groups of muscle parts called fasciculi. And then these fasciculi are also surrounded by that membrane. And then within the fasciculi are muscle fibers, again surrounded by membranes. And then the muscle fibers are made of these myofibrils. Myo means muscle, and fibril means fiber. And you could have hundreds or thousands of these little myofibrils in each muscle fiber. And then these myofibrils are going to be broken down even further to what's called sarcomeres. And in the sarcomere is where the contractile elements of actin and myosin are going to be. And it's the actin and myosin that actually is going to be the most functional or the smallest functional unit of a muscle where the muscle contractions are going to occur. And then you'll have a muscle fiber that'll like be really long, the length of the muscle, and you'll have these sarcomeres that are kind of lined up next to each other. So this series of actin and myosin relationships. So what is a sarcomere? A sarcomere, let's go back to this. Why is it doing that? I don't want that to occur. You know what I'm going to do? Give me 1 second. I'm going to open this PowerPoint up on my external hard drive, so I don't have to deal with that anymore. Give me 1 second. I don't need to do it on our hard drive. 1 second, everybody. This will be easier. Better. Okay, so if you look here, this is a sarcomere, all right? You see this little crisscross right here? Looks like a Z. This is a Z disc. All right, this is another Z disc, and in between the Z discs is your sarcomere. This is a sarcomere. All right, now you see this is a big bundle of these sarcomeres. This is called a myofibril. And these sarcomeres here are within the muscle fiber itself. This is the outer layer of the muscle fiber where the blood flow is going to be. These are called a sarcoplasmic reticulum. This is where calcium is. And these are where the calcium leaves the sarcoplasmic reticulum, and the calcium is going to seep into the receptors on the tissue of these muscle cells here. And right here is mitochondria. I don't know if you've ever heard of mitochondria. We're going to get into these next week. And mitochondria is where ATP is going to be broken down into energy. Sorry, where glucose is going to be broken down into energy in the presence of oxygen. If there's no oxygen, then it's an anaerobic breakdown of glucose and fat and protein, for that matter. And that will happen outside the mitochondria within the cell itself. But right now, we're just going to talk about these sarcomeres. And these sarcomeres are comprised of thin actin filaments. So these long, thin filaments here, actin, the red ones. And then the thicker, and you can't really tell from this, it's just a drawing, but the thicker filaments, the blue filaments, are going to be myosin. And on the myosin are these little like globules, these little heads that attach to the actin. And pretty much when a contraction happens, you see there's a space right here. There's also a space between the Z disc here and this actin filament here. So there's a space here. Eventually, when there's a contraction, the actin and myosin are going to slide against each other, and it's going to fill up these gaps. But you'll notice there's another gap here, there's another gap here, there's a gap here, and all the way down the muscle fiber, there are these gaps. And when those gaps are reduced, that's pretty much what a muscle contraction is. All right, so how are those gaps reduced? How does that happen? So that's what a sarcomere is. It's this basic contractile element of a skeletal muscle. So let's take a look at this, all right? An A band are dark stripes, I bands, light stripes. I've never really needed this information too much at all, but I'll show you what that means, all right? So if you take a look at this is another example of what, or another picture of a sarcomere. So they take this and they blow it up. So the distance between this line here and this line here is called the sarcomere, pretty much, right? This line here, this M line, is right in half, or cuts the myosin heads. These blue part is the myosin. It's the middle of the myosin, so that's what an M line is. An A band is the range that includes the outer edge of the myosin on either side. So this is an A band. And in the A band is a combination of the myosin, which is in blue, but you'll also notice that the A band includes some of the actin filaments as well. So the A band is the range that includes the actin and myosin. This is called an I band. An I band is only including the actin filaments. And the edge of the actin filaments, or the middle of the I band, is called a Z disc. This disc goes all the way around this myofibril. The reason it's called a Z disc is because it kind of looks like a Z. And the Z disc is where the actin filaments connect to each other. Then the Z disc is only made of actin, or is the range where there's only actin. Likewise, the H zone here is this middle portion here, not the M line. The M line is the actual middle line, the midline of the myosin. But the H zone is the region that only includes myosin. It's in the A band, but it would be the part of the A band that doesn't include any of the actin. So it would be just this middle part. I've never really needed that information in my entire life, but at least that is going a little bit further in regards to the structure of this sarcomere. The boundary between each sarcomere is the Z disc. So let's go into these protein filaments. Now, we'll talk about this a little bit later, but a protein filament is actin and myosin. So how does protein grow? Well, some people think that when they start to do resistance training or start lifting weights, their muscles will get bigger. It's not necessarily always the case, though. Like if you don't have enough fuel, and we'll talk about bioenergetics and protein and carbohydrate and fat. We'll talk about that next week. But like if you don't have enough fuel in your body, then you don't have enough resources to even make more protein. Like you could think about protein as like the metal. If you're going to make a building, you need like structure, you need metal, you need scaffolding, you need concrete, you need, sheetrock. protein is pretty much the metal and sheetrock and the thing that makes your body actually have structure to it. In order for your actin and myosin filaments to grow, or for your muscles to grow, or another word for muscle growth is hypertrophy, in order for that to occur, there needs to be like a specific recipe of strength training, of lifting, of exercise. Pretty much what's going to happen when you do exercise, especially at a particular intensity, these actin and myosin filaments are going to break. They're going to break down. It's called micro damage, micro tears. And when that happens, your body has to fix the building. So what's going to happen is your body is going to send out hormones, and we'll talk about hormones and muscles and exercise in, I think, 3 weeks. But in any event, for males, what happens is testosterone is sent from the endocrine system, and that testosterone gets to these nuclei here. And the testosterone gets into the nuclei, and that testosterone is going to seep into the DNA or the RNA that's inside of the nucleus. And when that happens, there's going to be mutations, and the mutations are increased actin and myosin protein synthesis. So when you're getting bigger and your muscles are growing, it's actually what's happening is these actin and myosin filaments are growing, and they grow like tree trunks, right? There's more and more actin, and they kind of like layer over each other. In any event, the actin and myosin filaments are the proteins that are involved in this sliding filament theory. We talked about these H zones and I bands and such, so we'll move on. First, we're going to talk about the myosin filaments. And you could think about the myosin filaments as the thick filaments. So if I look at this slide right over here, it's the myosin filaments would be these thick blue ones. Sometimes you can remember that because they're called myosin heads. And you can see that there are these little globule heads on the end of the myosin. And you can see on this side of the myosin filament, you can see that there are these little heads that have these little cross bridges here. And right now, this cross bridge is in this direction. And on this side of the M line, all of the cross bridge are going away from the M line. And on this side of the M line, it's opposite. The cross bridges are in this direction. And what's going to pretty much happen in a contraction is right now you can see that these myosin heads are not connected to this actin filament, but when they do touch each other, this cross bridge is going to switch from being in this direction to this direction, and it's going to pull the actin towards the middle, towards the M line, towards the middle line of the sarcomere, and all of these gaps here are going to be reduced. And because of that, the muscle is going to get shorter. But let's keep going with the myosin filaments. Now, these myosin filaments are intertwined with these globular heads, and the globular heads are going to protrude or stick out of the myosin filaments 360 degrees in that myofibril. So you could imagine this is 360 degrees here. And you're going to have myosin heads pointing out in all directions. All right, so that's what that 360 degrees mean. And it's these globular heads that are going to interact with actin filaments to make a contraction happen. Now, one of the things that we talked about, and we'll talk about later on today, is that muscle has an active but also a passive component. It has an active and a passive ability to store energy, like an elastic band. Right? If you can look at my screen, if I take this elastic band, all right, and I pull out, I'm going to get rid of my virtual background so it's easier to see. All right, so if I take this rubber band here, it's dead. It's definitely dead. It has no active component. It's not alive. There's no energy in this. It doesn't have any ATP. But it can create a tension, right? A tension enough so I can store energy, and I could use that energy for movement. Boom, and I fling it, and it flies across the room. So I can store energy in something that's dead or something that's not alive. But similar to storing, this is called passive energy, because this rubber band has no ability to actively make a contraction or store energy in itself. I'm pulling and creating tension and storing elastic energy in the material, and the material can use that energy for some reason. Boom. Now, just like this rubber band, a muscle has a passive component to it. It has a passive ability to store energy. So if I took my bicep off of my arm and I like held my bicep like this, I could stretch my bicep. I could pull on it and create tension in my bicep. All right, because a bicep has, and I'm going to also go like this 1 second. I'll do that later. Because your bicep and your muscles have this passive ability to store energy. So let's go back to these slides. And if you look over here, it says that this myosin filament is stabilized by titin. What that means is that there's a filament within the myosin. You'll see it's right here. It's called titin, and it kind of looks like a spring would look, like winding on itself. And that's exactly what it is. It stores energy. It's where the elastic energy or the passive component of muscle comes from. The ability to stretch and store energy like a rubber band. All right, so these myosin filaments are connected to titin, and you'll see that that's also, the titin is also kind of running through the actin filaments. So this is like a spring, and the myosin and actin combine to make an active component. So here's the passive component part of muscle and the active component. All right. Now you have actin. Actin is going to be a combination of 3 different proteins. So on the actin is a site where myosin can connect to. And when the muscle is at rest, right now my muscle is not contracting, there's tropomyosin, and tropomyosin covers these spots on the actin filament that myosin is going to bind to. So it can't really bind to it while it's at rest, and that's why there's no contraction. But also attached to the actin filament is this stuff called troponin. And what troponin does is it's going to move tropomyosin off of the myosin binding site so that the actin can touch the myosin. And when the actin and myosin interact with each other, in the presence of ATP and water, there's going to be a change in the angle of that myosin crossbridge that I showed you earlier. And when that angle changes, what happens is the sarcomere gets shorter. And when that sarcomere gets shorter, and all of the rest of the sarcomeres get shorter along the entire length of the muscle fiber, that's what a contraction, and that I should clarify, that's what a concentric muscle contraction is going to be. Now, there is many different types of muscle contractions. Later on, we'll talk about static and dynamic muscle contractions and concentric and eccentric muscle contractions, and what the real reason for concentric and eccentric muscle contractions is, but we'll get to that in a bit. So that's what actin and myosin is. If you've ever eaten a steak, you've eaten plenty of them. All right, so now let's take a look what a motor unit is. All right, a motor unit is an alpha motor neuron, And the alpha motor neuron is the nerve that's coming from the brain. And that alpha motor neuron is going to travel down a portion of the spinal cord, and it's going to branch out, and eventually that alpha motor neuron is going to go and innervate or connect to a bunch of different muscle fibers. You could have very large alpha motor neurons, and you could have very small alpha motor neurons. Usually when you're doing a task or a movement, the smaller alpha motor neurons fire first, and then sequentially bigger and bigger and bigger alpha motor neurons or bigger and bigger muscle or motor units are going to be innervated. So a motor unit is an alpha motor neuron and all of the muscle fibers it connects to. So you could have, this is a dendrite or axon in the brain, and the action potential is going to emerge somewhere, or we could even say that the brain creates this action potential mysteriously, not sure how. All right, not sure where they come from, but the brain is going to send an action potential. Yeah, I'll even go into it one more time. Like, if you're going to have a town, and the town has a bunch of different houses, all of those houses are going to need electricity, and you have this one department in the middle of the town called the electrical department, and for sure that electrical department is going to provide energy and electricity so that all of these houses could run their phones and their lamps and things like refrigerators and stuff. And sure enough, if the electrical department had a problem and it couldn't send out electricity to the town would sure have a problem functioning. But that's not to say that the electrical department is the brain of the town, right? If the plumbing of the town didn't work, or if the police department didn't work, or the fire department didn't work, the town would also have a problem functioning. So it's not like the electrical department, or in this case, the brain, is the sole thing that makes the body function. It's part of it. When you think of the brain as the sole thing that makes it work, that's called hierarchical motor programming theory. When you think about the brain and the heart and the lungs and the spleen and the liver and the kidneys and the skin and all your cardiovascular system, if I was to take the cardiovascular system out, the body wouldn't work. If I took the lungs out, the body wouldn't work. Or the digestive system, the lungs wouldn't work. The body wouldn't work. So what heterarchical, as opposed to hierarchical motor programming theory, says that all of the systems of the body function together to allow this body to move. Same thing, all of the departments in this town are functioning together to allow for motion to occur, not just one department. All right, hierarchical motor programming theory says this one department, the brain, sends all of the energy, and the electrical department doesn't even send or make the energy. Usually the energy is going to be made by like turbines and hydroelectric power and things. So where the energy comes from, so that the action potential, which is electricity, can be sent down the alpha motor neuron and into the muscle fibers itself. Arguably that's just from food, from calories. So you could say the calories run the body. If you don't have any calories, if you don't eat any food, the body's going to break down too, right? So, and then food that we'll talk about next week, food is just chemistry. It's just chemicals. So it could be the chemicals are the brain of the body. In any event, you just want to kind of rethink this whole process here. According to hierarchical motor programming theory, the brain sends this electrical signal, which is an action potential. That action potential is going to travel down your alpha motor neuron, and eventually it's going to get to the synapses of that neuron, and that neuron is going to innervate 1, 10, 100, 1,000 muscle fibers, not likely one. And then there's these neuromuscular transmitters, things that transmit the action potential or the electrical energy from the alpha motor neuron into the muscle fiber itself, into the receptors of the muscle fiber. Typically, those neuromuscular transmitters are acetylcholine and dopamine. Now, acetylcholine is the thing that makes this whole thing occur, it's like an exciting neurotransmitter. And the thing that works opposite of acetylcholine is dopamine. And an individual who has Parkinson's disease has a deterioration of a part in the brain called the basal ganglia. And what the basal ganglia does is it makes dopamine. So imagine if you had a lot of this acetylcholine, and we'll get into, let's go like this. Let's just finish this, and then I'll get into that discussion in a second. So the electrical energy, the action potential, travels down the alpha motor neuron, and it gets into this muscle fiber. Typically, what happens is the action potential starts in the brain, and this is a process called the excitation contraction coupling. Coupling is also another word for constraint or connecting one thing to another or a system. In the discussion of excitation contraction systems or coupling, it starts with number one, the action potential starts in the brain. I mean, you gotta question that in the first place, but let's assume it does. How it starts in the brain, don't know, but the action potential is going to travel down this alpha motor neuron. It's eventually going to get to the terminal branches, and the action potential is going to release acetylcholine. Acetylcholine is going to excite the muscle fiber. All right, the acetylcholine is going to cross that neuromuscular synapse, and it's going to bind to acetylcholine receptors on the muscle fiber itself, on the plasmalemma, the outside layer of the muscle fiber. Then that action potential is going to travel down the plasmalemma. It's going to get to the T-tubules. And when it gets to the T-tubules, it's going to trigger a release of calcium. That calcium is going to move the troponin and the tropomyosin so that the actin can connect to the myosin. And when that happens, especially in the presence of adenosine triphosphate, ATP, and water, all right, there's going to be a release of energy, and that release of energy is going to result in a muscle contraction. So that's kind of like the steps of what's going on. So we'll take a look at this figure here. Now we'll just talk a little bit about this acetylcholine. Acetylcholine is like, so this is your neuron. This is the neuromuscular or the synaptic cleft. This is the synapse between the motor neuron and the plasmalemma, the outside layer of the muscle fiber. And you'll notice on the muscle fiber, on the plasmalemma, there are these little purple things. Maybe if I can make it even bigger. You'll see these purple things here have this little shape to them. This is just a drawing. It's just a model, so I couldn't say that this is exactly what it looks like or whether or not they're purple. But you'll see this purple thing here has this little roundness cut out at the end. That's called a receptor, and the acetylcholine is going to be released from your storage vesicles at the end of the axon or the axon terminal. The acetylcholine is going to be released. It's going to go across the synaptic cleft, and that acetylcholine is going to be accepted by one of these receptors. And when the acetylcholine is accepted by the receptor, there's going to be a propagation of that actin and the action potential into the muscle fiber, into the sarcoplasmic reticulum. And when that action potential gets into the sarcoplasmic reticulum, what it's going to do is it's going to make it so that the troponin and tropomyosin intertwine so that the myosin heads can connect to the actin sites where the myosin heads are going to connect to. Now, what we were talking about with dopamine is what dopamine does is it releases so that this process stops. So somebody who has Parkinson's disease doesn't have enough dopamine. So they have too much of this acetylcholine flooding through their muscle fibers. And because of that, there's going to be too much contractile activity in the muscle fiber. And that's why you'll have somebody who has Parkinson's disease will have like a tremor, because there's too much motion going on, too much muscle contraction going on. So what do they do for an individual who has, or who potentially has Parkinson's disease? Is what they do is they make synthetic dopamine called levodopa, and that individual takes that as a medication. And also that levodopa, that dopamine, just like the acetylcholine has to have receptors, the medication with the dopamine in it also has a different type of medication that's going to help dopamine attach to the dopamine receptors. So you'll have different types of Parkinson's medication, different brands, and the different brands are a combination of the levodopa, the pretend dopa, and receptor proteins that are going to allow that levodopa to connect to the plasmalemma and to the receptor sites more readily. And different medications are different percentages or concentrations of levodopa compared to its receptor. And so that's one of the things that you can use to reduce muscle stiffness and tremors associated with Parkinson's disease. But in any event, when the acetylcholine gets into this receptor site, it's going to trigger a release of calcium from the T tubules, and that calcium is going to attach to the troponin, and the troponin is going to move away from the tropomyosin and allow for these myosin heads to attach to the actin. All right. So what does calcium do? Well, when calcium right now is housed in the sarcoplasmic reticulum, and eventually it's going to go through these tubules and it's going to seep into the muscle fiber itself. And the sarcoplasmic reticulum is very sensitive to the action potential, to electricity. And once the electricity arrives at the sarcoplasmic reticulum, it's going to flood the sarcoplasm. It's going to flood, the sarcomere with calcium. That's going to allow for the tropomyosin to move away from the myosin binding site and allow actin to connect to it. So this troponin and calcium combination, see this little line right here? Troponin, calcium complex. Another word for complex is system. And what happens is this system between troponin and calcium allows for the tropomyosin to move so that myosin and actin can connect to each other so a contraction can happen. And that is pretty much the basis of this theory here, this theory, not fact, this sliding filament theory, how muscles create movement. It says, this is a theory, but this is how it happens, right? It's like talking out both sides of the mouth here, right? The sliding filament theory is theoretical because we have no idea how the action potential starts in the brain, when it starts in the brain, where or how the brain does it. If anybody can explain that, right, you'll pretty much have the universal idea for how motion occurs in the human body. But for right now, we'll just agree that it's a sliding filament theory. The filaments, the actin and myosin, slide past each other. And when they do, the muscle gets smaller. In the relaxed state, these actin and myosin interactions don't happen because the troponin and tropomyosin stop that from happening. All right, and you'll see this says myofilaments overlap a little bit. What does that mean? Well, you'll see in the A band, here, you'll see that there's a little bit of an overlap between the myosin heads and the actin filament. There's a little bit of an overlap in this zone here. All right. And when the muscle contracts more and more, the actin filament is going to move in this direction. There's going to be more and more and more overlap until this actin filament hits right here, hits in the middle. And then the muscle can't contract any further. When the muscle is overextended further and further apart, what happens is this overlap becomes smaller and smaller. And right now, let's just to count. Right now, you have about 1, 2, 3, 4, 5 myosin heads can interact with the actin filament. But imagine if this spread out. Now, if the muscle is more contracted, you'll have more myosin heads can interact with more of the actin filament. That's going to result in a more forceful contraction. When the muscle is overextended too far, extended too far, only a few of these myosin heads can even interact with the actin filament. So you're not going to be able to make such a forceful contraction. So a forceful contraction has something to do with the number of myosin heads that can interact with the actin filament. It's like you're playing tug of war, and right now you have 5 people pulling in this direction and 5 people pulling in that direction. But in this situation, you have 10 people pulling on the rope. So there's going to be a better contraction here. We'll talk about that later. In a relaxed state, there's no actin and myosin interaction at the binding site because tropomyosin is covering the binding site. And the myofilaments, the muscle fiber filaments, or the actin and myosin, only overlap a bit. When the muscle contracts, the myosin is going to pull the actin towards the middle of that Z disc, the M line rather. That's called the power stroke. And I'll show you another slide that has a bit of a step-by-step description of that. And the filaments slide past each other, sliding filament theory. And then that causes the sarcomeres and the muscle fiber and the entire muscle to shorten. Now, when a muscle fiber shortens, that's called a concentric muscle contraction, getting smaller with the middle, concentric. But there's also eccentric muscle contractions, and we'll talk about that in a bit. All right, so this is a muscle in its relaxed state, a little bit of overlap. And as the muscle contracts, there's more and more overlap. There's more and more actin and myosin interaction, so you can produce a more forceful contraction. All right, now, see these I'll wait until I get to this slide over here. So after the change in direction of the myosin cross bridge, after that motion occurs, the myosin is going to detach from where it is on the actin. It's going to move to a different actin binding site and pull further and pull further. So there's this never-ending cycle of attaching myosin to actin. The power stroke happens, myosin detaches, attaches to another spot in the actin filament, and pulls again so that the muscle can get smaller and smaller and smaller. But this power stroke, all right, in order for a light bulb to have power, it needs energy. In order for a car to have power, it needs gasoline. In order for the myosin to change its angle of orientation and detach, the myosin is going to be moving. Either the myosin cross bridge is going to move in this direction, or it's going to release and move in the opposite direction. And movement or power requires energy. And that energy is going to come from ATP. Now, this process of the myosin detaching, attaching to a new active site down the actin is going to happen until the muscle stops contracting. or the muscle can't contract anymore. Now, if I take my arm and I flex my bicep all the way, there's complete 100% overlap of my actin and myosin, and the Z discs have reached the myosin filament. I just cannot contract my muscle any further. So there's no place for the myosin to attach further down the actin filament. There's just no place for it. So the contraction will stop. That's when my muscle is completely contracted. Or the muscle contraction is going to stop when there's no more action potential. And then when that happens, calcium is sent back into the sarcoplasmic reticulum. One of the things that helps with this is magnesium. So sometimes if you get, or my athletes get muscle cramps, then what I'll suggest to them is take a multivitamin that has magnesium in it, because magnesium helps the reuptake of calcium back into the T-tubules and the sarcoplasmic reticulum. All right, so when this stops, the myosin is going to return back to its original position, but again, that's going to require energy. And if there's no ATP, then the myosin can't detach and it can't move back to its original position. And why would there be no ATP? Well, if I was dead and I wasn't regenerating any ATP, then what would happen is the myosin would get stuck in its final position and it wouldn't be able to relax the muscle. And that's what rigor mortis is. All right. So let's take a look at this slide here, and I'll try to like pull up each one of these pictures for you. So this is in the beginning. This is in the beginning, all right? The myosin filament has this cross bridge, and you'll notice the cross bridge is angled in this direction, right? This is like a 45-degree here, and this is where ATP can bind to the myosin. Now, this myosin right now is attached to your actin molecule at, say, this binding site number 1. Now it's ready to contract. And what's going to happen is this cross bridge eventually is going to move in this direction and attach to number 2 and move back in that direction. Then it's going to attach to 3 and move, and attach to 4 and move. And it's going to take this actin molecule and pull it along the line that way. And this actin filament is going to slide past this myosin filament, the sliding filament theory. So how does that happen? Well, in order for this cross bridge to move at all, there's going to have to be energy. And ATP is floating around in the bloodstream. There could be the case that there's ADP, adenosine diphosphate, and phosphate molecules that are floating around the bloodstream that are being recycled. And when they're recycled to ATP, or when ATP is created through glycolysis or the phosphocreatine system, we'll get into that next week, or aerobic glycolysis in the presence of oxygen, all right, ATP is going to bind to this ATP binding site right here at the myosin head. Now, when ATP binds, what happens is the myosin head now detaches from the actin filament. The ATP is going to be released, or energy in that ATP is going to be released through hydrolysis. Hydro is water, and lysis. Anytime a cell lyses, it breaks in half. So water is going to be broken in half. That's why you need water when you exercise or when you get thirsty, because this whole process of breaking down ATP requires water. So either you're going to get it from the tissues of your cells, or you're going to replenish the water in your body by drinking water. So you need to be relatively quenched of your thirst. You need to have water in your system in order for ATP to break down. Water is going to break in half, and when the water breaks in half, it's going to bust this ATP into adenosine diphosphate and a phosphate ion. And that's going to release energy. All right. When that energy is released, that energy is going to be used, and that energy is going to be used to take this angle here of this myosin cross bridge and move the cross bridge down the actin filament so that the myosin and actin can attach at a different attachment site. So right now it's going to go from attachment site number 1, and eventually it's going to move to attachment site number 2. So the ADP is going to be released. Sorry, the ATP is going to be broken down into ADP and your phosphate ion. There's going to be a release of energy, and that release of energy is the gasoline for this particular motion to occur. Now, this cross bridge is going to change its angle of orientation. Now, this drawing says it goes from 45 degrees to 90 degrees, but again, that's just a drawing. But when it does go to 90 degrees, it's like you can see now this cross bridge is now pretty much vertical, whereas before it was 45 degrees. This motion takes energy. The energy comes from the breakdown of ATP. And now this myosin head swings in this direction. And instead of being over the actin binding site number 1, it's now over the actin binding site number 2. And now here comes the power stroke, all right? The power stroke is where this myosin head and the cross bridge move back to that 45-degree angle, but instead of being attached to the binding site 1, it's attached to binding site 2. So it's going to take this actin filament and move it down the line. It's going to slide the actin filament down. This is going to happen until this calcium is pumped back into the actin, the sarcoplasmic reticulum. For the contraction to happen more and more and more, once the contraction gets to this particular point, there's going to be more ATP. The ATP is going to be broken down again into ADP, and there's going to be another release of energy, which is going to take the myosin head, attach it to number 3, and move it again. And then more ATP, another detachment, and the myosin head will attach to 4, and then to 5, until it reaches the end of the Z disc. And when it does, there's no more opportunity for the actin filament to move. The muscle is at complete contraction, and then the contraction is going to end. All right, so that's pretty much how a muscle is going to contract, at least according to the sliding filament theory. Okay, so the energy for all of this contraction comes from adenosine triphosphate. It's an adenine group with 3 phosphates connected to it. And this phosphate group, or one of these phosphate groups, when it gets ripped off, is going to release energy. And that energy is going to be immediately used for the myosin head to change its angle of orientation. All right, ATPase is an enzyme that's present in your muscle fibers to allow for this to happen. Anytime you see ase, it implies that this is an enzyme. All right, that's the suffix for enzymes. So this ATPase is going to be present in your muscle fibers, but depending upon the muscle fiber, there could be more or less ATPase. Now, a slow twitch muscle fiber, a muscle fiber that is involved with endurance running and not power, is going to have less ATPase because it doesn't have to contract as fast. So it doesn't have to recycle the ATP next to the myosin fast enough. So one of the big differences between type 1 and type 2 muscle fibers is that type 1, or slow twitch muscle fibers, they don't have as much ATPase as type 2 muscle fiber types. Because type 2 muscle fiber types have more of that enzyme that breaks down ATP, it can create this process. This process can happen faster and faster and create more power. So that's one of the big differences between type 2 and type 1 muscle fiber types, but we'll get into that in a couple minutes. So when the muscle relaxes, the action potential is no longer propagating down the alpha motor neuron. The sarcoplasmic reticulum stops sending calcium into the T tubules and stops the interaction of troponin and tropomyosin, and makes it so that the actin and myosin can no longer connect at the binding site. So magnesium helps this calcium get sucked back into the sarcoplasmic reticulum, and it's stored in the sarcoplasmic reticulum until another action potential arrives. All right. All right, so let's move on to the next section, which is the part about muscle fiber types. Now, there's different types of muscle fiber types, and you've all experienced this. I particularly like eating white meat chicken, white meat turkey, all right? But my dad always loved dark meat. So where does that come from? White meat is lighter in color because it doesn't have as much hemoglobin, and hemoglobin is red. Dark meat has lots of hemoglobin. So what is the difference between white meat and dark meat? Well, let's talk about a duck. All right, a duck has lots of dark meat. Actually, a duck I don't think has any white meat. It has all dark meat. Why is that the case when a chicken has white meat? Well, ducks migrate, and when they migrate, they need to fly for a long period of time. Therefore, a duck is going to have lots of type 1 muscle fibers, especially in the wings, where those wings need to flap and flap and flap and flap and flap for a long period of time. But chickens don't fly. So chickens don't have too much dark meat. They don't have too much type 1 slow twitch muscle fibers in their chest. In fact, they run a lot. They run really fast and they run a long time, and they're always walking around. So a chicken is typically going to have type 1 muscle fiber types in its legs and its thighs so that it can run fast away from you. But a duck has to swim a lot, and a duck has to migrate. So a duck typically has more type 2 muscle fiber types. Sorry. A duck has to swim and fly a lot. So those muscles are going to need lots of hemoglobin and lots of energy. So typically, I made a mistake, a duck is going to have more dark meat, more muscle fiber types that have hemoglobin, therefore more type 1 muscle fiber types. So type 1 muscle fiber types are slow twitch muscle fiber types. In fact, that's what they used to call them, but they don't really call them slow twitch anymore. They call them type 1 muscle fiber types. Typically, there's a relatively, I guess, even proportion of type 1 and type 2 muscle fiber types in an individual, but the percentage of type 1 and type 2 muscle fiber types from person to person might change. For instance, there are athletes that I have in the gym that have much longer, thinner, leaner muscles. And it seems like they're able to stay on pommel horse or on parallel bars for a long time without getting tired. But they never really get much bulk. They never really hypertrophy too much. But then you have other people who have like smaller, more compact, tighter muscles. They're able to go through hypertrophy much easier, and they're much stronger on rings. They can create a huge amount of speed and power on vault. So type 1 muscle fiber types are slow twitch muscle fiber types used for endurance training, marathon runners, long distance running, long distance rowers, swimmers will typically have a higher concentration of type 1 muscle fiber types. And it takes much longer for those type 1 muscle fiber types to reach their peak ability to create force. Tension in a muscle is also force. It takes about 110 milliseconds for these type 1 muscle fiber types to allow all of this stuff to occur. Type 2 muscle fiber types create force much faster. One of the reasons why I mentioned earlier is because they have more ATPase. They also potentially have more ATP binding sites, and so their ability to create power happens much faster. But there's different types of type 2 muscle fiber types. There's different types of these fast twitch muscle fiber types. There seems to be only one of these type 1 muscle fiber types. When you do exercise, It's very seldom at all, if it ever occurs, that you can switch from type 1 to type 2 muscle fiber types. And the other thing I should mention too is when you're born, you have a particular number of muscle fibers. And for the most part, that number of muscle fibers stays that number for your whole life. So when you do hypertrophy training, you don't increase the number of fibers. What you do is you increase the diameter of the fibers. Like a tree grows, but it doesn't grow 5, 6, 7 trunks. It stays one trunk, but that trunk gets thicker in diameter as it grows. So type 1 muscle fiber types are oxygenated, have more hemoglobin, are typically darker in color, and type 2 muscle fiber types are used for power, explosive power, and are typically going to have more ATPase. Type 1 muscle fiber types, when you exercise, and these type 1 muscle fiber types, they change because of exercise. Typically, the changes that are going to happen to type 1 muscle fiber types are changes that are going to allow them to utilize oxygen better. So there's going to be an increase in the number of mitochondria. There's going to be an increase in the number of blood vessels that course through the epimysium, endomysium, and paramysium. There's going to be, in type 2 muscle fiber types, however, there's going to be other adaptations, like the increase in the amount of ATPase or other adaptations that are going to make it so that they can make a more forceful contraction. All right, this is a slide that is that you have some muscle fibers that are darker. They're stained with a particular stain, and you'll see that these muscle fiber types that are dark are going to be your type 1 muscle fiber types that have a large amount of hemoglobin. That's why they're dark color. And then you have other ones that are a little bit whiter. These are going to be your type 2 muscle fiber types, and then others that are kind of gray, that have a little bit more of the ability to these. So these ones, the gray ones, are still type 2 muscle fiber types, but they're type 2X muscle fiber types. The reason they're type 2X muscle fiber types is because those are fast twitch muscle fiber types that have a little bit more ability to oxygenate and use oxygen as fuel. So they're kind of stained as gray. All right, so in type 1 versus type 2 muscle fiber types, in type 2 muscle fiber types, fast twitch muscle fiber types, they have an ATPase that allows for faster contraction cycling. What that means is faster contraction cycling. Now, let's go back to this slide over here. In order for the actin and myosin, for the myosin to change from the binding site number 1 to number 2, there needs to be a release in ATP. The ATP needs to be broken down into ADP, and that requires this ATPase to do that. Then what happens is the myosin head attaches to the actin, it slides down, it changes its angle of orientation rather, it slides down, but then there's going to be another release of ADP so that it can attach to number 3 and then attach to number 4. And ATPase is going to make this ADP break down into energy so that that cross bridge can change its angle of orientation. That's called recycling. All right, that's called this contraction cycling. And for fast twitch muscle fiber types, there's like a fast version of this ATPase that breaks down ATP much faster. So for type 2 muscle fiber types, there's a faster contraction cycling. How do you know whether or not your athlete has type 1 versus type 2 muscle fiber types? Well, I really don't know, because I haven't done any muscle biopsies in my gym because I don't know how to do that. And I think that's probably somewhat invasive. But I can take a look and use my best guesstimate. And I find that there are some athletes that have longer, thinner muscles that don't ever seem to get tired when we do our warm-up. And then there are other athletes that are super powerful, super strong. They go through hypertrophy training. Like we do a pretty extensive strength and conditioning program at the gym. And some kids will put on, like one kid put on, I mean, he's 11 years old and put on 20 pounds last year. You know, he got big. There's other kids though that do the same exact strength and conditioning program. They'll put on 10 pounds. That could be because of nutrition, but it could also be because that one kid who put on a good amount of muscle mass has predominantly type 2 fast twitch muscle fiber types. Really good on vault, really good at being explosive, but not very good if I said, I want you to do, I want you to run 2.5 miles. As a gymnast, I was not very good with running. I did a lot of power stuff, but not running. And for the most part, I believe that I have a higher percentage of type 2 muscle fiber types than type 1 muscle fiber types. So in order to tell, you could do a muscle biopsy, which is take a little slice of the muscle and then you take a look at it under the microscope. Or what you could do is you could do electrophoresis, which is setting up these gels and let electricity separate the sizes of the myosin that is different in type 1 versus type 2 muscle fiber types. But so there are ways of telling, but for me it's difficult because I don't really have the ability to use those things.
Can I ask a question?
Yeah, of course. Go for it. What's up? Yeah.
So when you were talking about slow twitch versus fast twitch and I guess the efficiency of using oxygen, does that oxygen part come into play with how quickly the ATPases are like, is that where the oxygen comes into play with the ATPases, or where is that in the contraction that?
No, I think that, I mean, I don't know. I'm not 100% sure about how now oxygen is going to start interacting with ATPase. I probably have to do a little bit more reading and research on that. But I would say that the oxygen for your type 1 muscle fiber types is going to come into play because remember, type 1 muscle fiber types are going to be used for a longer period of time. And if that's the case, then you're going to need to break down a different substrate than ATP because ATP, there's not enough ATP in the body to really last for longer than 10 seconds of activity. So the oxygen is going to be now used to shuttle your pyruvate, which is your glucose, into the mitochondria. And when oxygen attaches to the pyruvate and brings it to the mitochondria can go through the Krebs cycle, and it can go through the electron transport chain and result in more ATP. So I would say that that's probably where the oxygen and type 1 muscle fiber types are going to interact a bit more.
Okay, thank you. That makes more sense.
Right, because ATP can break down in the muscle fiber without oxygen. That's going to be your fast glycolysis without oxygen. So I don't necessarily think it's 100% necessary for ATP to release its energy, especially if there's ATPase with oxygen. Oxygen is just going to make it so that when it finally does attach to the pyruvate and gets to that mitochondria, then rather than creating one explosion of ATP for 1 unit of ATP, in the presence of oxygen, that molecule of pyruvate is going to result in like 44-fold times more of the explosion of ATP, right? So I would say that it's not necessarily related to the breakdown of ATP, but it has more to do with production of more ATP. That's what I would say.
I also have a quick question.
Yeah, go for it.
So are you either like born with type 1 or type 2 muscle fibers and then it can change over time, or does it just, like if you build more muscle, can it change over time or does it just stay the same?
No, for the most part, the research that I have is there's very little research that suggests that you can switch from type 1 to type 2 muscle fiber types. Right? For the most part, it seems as though people are born with a particular percentage. Now, that's not to say that everybody's muscle has the same percentage of type 2 and type 1 muscle fiber types, but it seems as though that when you're born, you have a particular percentage of muscle fiber types throughout your body and in the muscles, and that doesn't seem to change too much. What changes with activity and exercise is things like more blood vessels, more mitochondria, faster release of calcium, increase in ATPase, things like that. But it doesn't seem to be with the research that there's significant change from type 1 to type 2 or vice versa. Now, there is research that suggests that type 2A and type 2X muscle fiber types can intertwine or interchange between each other. and the type 2X muscle fiber types utilize oxygen better. So it could be the case that with that switch in muscle fiber types, there's an increased ability for oxygenation in type 2 muscle fiber types, but doesn't seem like there's too much switch from type 1 to type 2 muscle fiber types. Okay, so other differences between type 1 and type 2 muscle fiber types. Type 2 muscle fiber types have a higher developed sarcoplasmic reticulum, and that means that you're going to be able to store and release calcium faster, which is going to make, this is the velocity of contraction. The velocity of contraction in a type 2 muscle fiber type is going to be 3 times to 5 times faster, which means a greater power stroke, more contraction power. So type 2 muscle fiber types are more powerful than type 1 muscle fiber types. And it's also the case that if you're gonna do a motion, a movement, usually the type 1 muscle fibers are gonna fire first, especially if the movement is very low intensity. Type 1 muscle fibers and motor units are usually innervated by smaller neurons. So it seems to be the case that there's like this order of excitation from smaller to larger. Larger motor units are going to require more energy, and usually the body wants to conserve energy. So the smaller motor units are going to fire first until the intensity of the activity is higher and higher, and the need for more power is there. So then slowly but surely more type 2 muscle fiber types and motor units that are innervating type 2 muscle fiber types. And again, motor units that are innervating smaller type 2 muscle fiber types are going to innervate sooner than larger motor units. So it's like from small to large. So the motor units in type 1 muscles are typically smaller, and therefore they're going to fire first, but not provide as much power. And because of that, it seems as though the peak power is for your type 2X muscle fiber types is going to be greater than your type 2A muscle fiber types. And those are all going to be greater than your type 1 muscle fiber types. All right, but that's going to be dependent upon the size of the motor units, the number of the diffusion or the complexity of the sarcoplasmic reticulum, and all sorts of other issues. All right, now every person is born with, nobody's the same. So everybody has slightly different ratios in type 2 and type 1 muscle fiber types. So for me to teach everybody in my gym or to train everybody in the gym the same way might actually not necessarily be effective or even possible. So especially age-wise, but let's just talk about muscle fiber types. So sometimes the kids, what I do every week, every Wednesday, today's Wednesday, so I'll do today, is I take everybody's body weight, their body composition, and their bone density. And we're doing all this strength and conditioning, especially during the summer when it's our hypertrophy phase in our periodization chart. Everybody does the same exercises for the most part, maybe different intensities given their age and their ability. And then sometimes the kids are looking at their weight, and one kid will put on 2 pounds a week and another kid will put on a half a pound a week. And they're always like trying to compare each other. And I find that not to necessarily be functional because everybody's physiology is different. Everybody has different ratios. Within an individual, it seems as though there's a similar ratio of type 1 to type 2 muscle fiber types from arms and legs. For individuals who usually do endurance, and it's not individuals who choose to do endurance, but over the course of your youth, you're exposed to many different sports, and typically kids will continue to do sports that they're good at. So a lot of times people will say, does gymnastics make people short? Does it make people bulky? No, but the kids who are short and bulky are typically good at gymnastics and want to continue to do it. So individuals who typically will gravitate towards endurance type activities are typically individuals who have higher number of type 1 muscle fiber types. And the same can be said for individuals who do power sports. Typically they'll have more type 2 muscle fiber types. And I think that the choice to do one sport versus another is a bit of an emergent property. Like, I don't ever recall making a decision to do gymnastics. I just did it. It just kind of was part of my life, and part of my life because probably the physiology is driving the success in the sport, and the success in the sport drives my interest in the sport. Therefore, the physiology drove the interest in the sport and allowed my gymnastics career to probably emerge from kind of like nowhere. But there are muscles in your body that are typically used by everybody to do the same thing. Like your soleus is predominantly used for walking. And because your lower leg is used predominantly for walking, typically there's going to be a higher percentage of type 1 muscle fiber types in your gastrosoleus unit for everybody. So everybody has different ratios. Some muscle comparisons can be similar, but then other muscles are top heavy in terms of certain muscle types. This just shows that type 2, we already just, we talked about this a little bit. Type 2 muscle fiber types can create more power than type 2A and type 1 muscle fiber types. Type 1 muscle fiber types during exercise are typically involved with aerobic endurance because they can utilize oxygen more effectively. There's more vascularization. Although there's less ATPase, usually there's more mitochondria, there's better vascularization, and there's a better ability to incorporate or to pull in oxygen and shuttle the oxygen to the mitochondria for activity. But typically, because of their physiology, type 1 muscle fiber types are not going to be used for very high intense activities. And the more and more intense the activity is, typically the more you're going to start to recruit your type 2 muscle fiber types. Now, because type 1 muscle fiber types are going to utilize oxygen more so than type 2 muscle fiber types, they're going to get energy differently. Type 2 muscle fiber types are going to get energy typically from either the phosphagen system or from fast glycolysis. And neither one of those are going to utilize carbohydrate to the extent that a type 1 muscle fiber type would use. So the energy, the ATP that a type 1 muscle fiber type is going to get is typically going to come from fat or carbohydrate stores. And if it's coming from fat, it's going to be the fat's going to be broken down from a triglyceride in beta oxidation and go into the mitochondria and break down eventually to through the Krebs cycle and result in ATP from that mechanism. And here, the carbohydrate, if your carbohydrate is a source of energy, it's also going to be broken down typically with oxygen in the Krebs cycle. Type 2 fibers, though, they're going to get energy anaerobically, so they're not going to be getting ATP from the Krebs cycle. They're going to get ATP from fast glycolysis, and we'll go over this next week, fast glycolysis and without the presence of oxygen. All right, and during exercise, type 2 muscle fiber types are going to create more force, but they're going to be used for high intensity endurance events. So they utilize oxygen a little bit better. They're going to fatigue faster than type 1 muscle fiber types, but type 2A muscle fiber types can use oxygen better than type 2X muscle fiber types. So individuals who are doing like 100-meter sprints, they'll typically be using more or have a higher percentage of type 2X muscle fiber types than individuals who are going to do a little bit of a longer, more intense run. All right, so what determines the type of fiber types that you have? Well, genetics. All right, and also the number or type of alpha motor neurons that are going to innervate those fibers. Now, here it says that training factors can induce small changes in fiber type, but the changes in fiber type that they're talking about are typically from type 2A to type 2X, not type 2 to type 1. And these changes, I think that Cassie asked that, the changes are so small that it's not necessarily going to result in a huge increase in sport performance in any manner. All right. Now, with aging, there's a reduction in type 2 motor units, which means that the number of muscle fibers that are innervated by this alpha motor neuron can decrease. But we'll get to that when we talk later on in the semester about exercise physiology and the effects of aging. All right. Let's talk a little bit about, let's move on to here. So now what we want to do is what are the muscles used for? All right, maybe we can have a little bit of a discussion here. In a car, you have ways of generating power, and you also have ways of dissipating power. All right, in a car, if you want to generate power, what you're going to do is you're going to press the gas pedal. And if you want to stop that power, what you're going to do is you're going to press the brake pedal. There's different things. There's a gas pedal over here and a brake pedal over here, and they do very different things. But in your body, you don't have a gas pedal and a brake pedal. You have muscles. And the muscles act as the gas pedal and the brake pedal. So there are some muscle contractions that are static, that don't move. Iso is the same, and metric is distance. So there are some muscle contractions that your muscle stays the same distance. That's like if I'm trying to hold up like a bag of rice with my arms. My arms are going to be at 90 degrees, and my arms are neither going to be extending nor will they be flexing. My muscles are certainly contracted, but my joint angle isn't changing, and there is no sliding of the filaments, although there is recycling of ATP. That's called an isometric, the same metric contraction. So there are static contractions. Static means not moving, not changing. And then there are dynamic contractions. And I think when we're talking about exercise physiology, we're probably going to be talking or considering mostly your dynamic muscle contractions. All right. In this case, a muscle is producing force, but it's not getting longer or shorter. And in this case, the muscle is producing force and changes length. Now, if you look at your book and you have an anatomy and physiology book, and it's telling you about origin, insertion, and contraction, origin, insertion, and action, origin, insertion, and action. Usually the action that is listed in an anatomy and physiology book is related to the concentric contraction of the muscle. But a muscle can not only shorten, but a muscle can produce force and lengthen. So a concentric muscle contraction with the center or towards the center, towards that M line. A concentric contraction is when all of the sarcomeres shorten. They get closer to the center. They go with the center of the muscle, concentric. And they're producing force. An eccentric muscle contraction is the opposite. Eccentric, I find like an eccentric person is somebody who's kind of like on the fringe. They're maybe somebody that, you know, I don't know, I don't want to say this in any derogatory manner, but in my imagination, they might not have as many friends because people can't relate to them. So people kind of like spread away from them. They go away from the center. They go away from that person. So concentric is shortening with the center, and eccentric is away from the center. In this case, the eccentric muscle contraction is producing force, but it's getting longer. So over here it says lowering a heavy weight. Like if I'm doing a bicep curl and the bicep is shortening and the weight is going away from the ground, then that's a concentric muscle contraction. And typically eccentric muscle contractions, the weight is going towards the ground. You're lowering a heavy weight. And right now we have gravity. Gravity is always pulling you downwards. All right, gravity is always pulling in this direction, downwards. Gravity's pulling downwards. All right, gravity's pulling in this direction. And for an eccentric muscle contraction, likewise, gravity is pulling in this direction. So these two contractions are very different. With a concentric contraction, you're usually lifting a weight. You're countering gravity. Gravity is pulling the weight down, but your arm is pulling the weight up. All right? And what you're doing when you do a concentric muscle contraction is you're overcoming the pull of gravity. With an isometric contraction, if, say for instance, gravity is pulling down with 10 units of force, and you're pulling up with 10 units of force, no motion is going to occur. And that's typically what's going to happen with an isometric contraction. But with a concentric contraction, the pull of gravity, and maybe what I should do is I should do this. The pull of gravity is smaller than the force produced by the muscle, and therefore the resultant motion is your limb or the weight going upwards away from the pull of gravity. That's usually what a concentric muscle contraction is. And if you think about going to the gym, you're working concentric strength. For the most part, that's against gravity's pull. And you could also think that a concentric muscle contraction is a contraction where what you're doing is you are generating power. So this is a generating power. You're generating, or your body is acting as a gas pedal. So you generate power. Usually the body is going to go against gravity. It's going to usually go from low to high. This is your body's gas pedal. Concentric muscle contractions are your gas pedal. An eccentric muscle contraction is usually lowering things down. Gravity, if I didn't have an eccentric muscle contraction when I'm lowering the weight down, the weight would free fall and it would snap my arm straight. Probably rupture my bicep. So usually an eccentric muscle contraction is overcoming the force of gravity, overcoming that drop. And because of that, rather than generating, it's absorbing power. So typically the eccentric muscle contractions, like in a car, absorb. In a car, you generate power with the gas pedal and you absorb power with the brake pedal. So typically in the body, the muscles are acting as the gas pedal and the brake pedal. Now we have an interesting thing that we could talk about. You know, some people think that injury happens because of overuse or because of lack of strength. But what happens if I generate power too long and I don't have the eccentric strength to absorb that power? What happens if I'm driving down the street, I'm pressing the gas pedal, all of a sudden I need to absorb the power in the car by pressing the brake? What happens if I'm on my phone texting while I'm driving and I try to absorb the power too late? What happens then? Drew, what happens then, my man?
I'm not sure. Can you repeat that?
Yeah, I'm driving in a car, I'm going down the road, I'm generating power. And the way to generate power in a car is by pressing the gas pedal. So I'm generating power. There's breaking down gasoline, there's firing in the pistons, there's movement. I'm generating power, but I'm on my phone and I'm texting and I have to brake, but I absorb, I brake, I press the brake pedal too late, like 3 seconds too late. What happens then?
You might crash.
I might crash. There's going to be too much power going through the system for too long a period of time. And if there's too much power going through the system, I could either hit the car in front of me, I could drive off the road, or I could, just like Drew says, have an accident. And your body is very similar. What happens if I don't have any brakes? Has anybody ever tried to push the brake pedal and they just don't have the ability to absorb the power because they don't have any brakes? What happens then? A crash, a big problem. Not good. So not only do you have to have enough gasoline in the car, enough nutrients to make the car go and to generate power, but you also have to have brake pads. You also have to have brake fluid so that you can absorb the power that you're generating. And if I cannot absorb the power because of strength of the gas pedal, the brake pedal, the functionality of the brake pedal, or because of the timing of when I apply the brake pedal, I could have an accident. So one of the things you want to consider is injuries occur not necessarily because your muscles aren't strong enough. It could occur because your muscles just don't have the eccentric capability to absorb the forces that are pushed through the muscle at any particular time. And because of that, let's say I'm throwing, all right, I'm a baseball player and I wind up, I store energy in my muscles, my anterior shoulder muscles fire concentrically to throw the ball forward. And the thing that's going to slow my arm down is either the tissues of my arm or the eccentric ability for my posterior shoulder muscles to fire and slow the motion down that I create through my windup and my concentric anterior shoulder muscle firing. So if I only do concentric muscle contraction strength as a baseball player, and I cannot fire eccentrically enough, what's going to happen is I'm going to throw my shoulder out of the socket. Or another example could be, what happens if I'm generating power in the same throw and my posterior shoulder muscles are strong enough eccentrically? What happens if they fire too late? What happens if the timing between the eccentric and concentric muscle contractions that generate and absorb power, what happens if the timing is disrupted between these two? Again, I'm not going to be able to absorb power fast enough. And then you're going to take all of that force and push it into your tissues. And then eventually the tissues are going to start to break down. If we have time today, we'll go through the stress strain curves. We have plenty of time. We'll do that. We'll go through stress strain curves of muscle, tendon, and ligament and show you what might happen to tendon, ligament, and muscle when there's too much force going through those tissues for too long a period of time. Typically, individuals who have rotator cuff problems, there's research out there. Like, why do you think that a little league baseball player shouldn't throw too many pitches during practice or throw too many curveballs? Well, because their muscles' ability to eccentrically fire and slow down that incredible speed of the throw just isn't there. Or what happens if they play spring ball, they play summer ball, and then they take 6 months off because now they're playing basketball and doing gymnastics, and they go back to their spring practice, and the very first day, the coach says, okay, let's do long toss. Well, the timing between this generating and absorbing power in their shoulder concentrically and eccentrically is probably disrupted. And now they're not strong enough or don't have the appropriate sequencing of their muscles to slow down and absorb all of this generated power. And now their tissues are going to have to absorb the power for them as opposed to the eccentric muscle contraction. if I can't slow things down eccentrically, then the stretch and the tension of the tissue itself is going to be the thing that slows down this generated power. And that's going to start to cause the rotator cuff to tear slowly but surely until eventually there's a rotator cuff tear. So when you go to the gym, not only do you want to work concentric muscle contractions going away from the ground, bicep curls, but you also want to do your negatives. You don't only want to do a chin up, but you want to lower down slowly so that your muscles have an opportunity to learn how to absorb power and not just generate power, because your muscles do both things. They absorb and generate power. They are the gas pedal and the brake pedal. And if I have a functioning gas pedal, but my brake pedal doesn't work, I can't do anything. Or if I have a great brake pedal, but my gas pedal is broken or I don't have any gasoline, I also can't do anything. So in the next couple of slides, we're going to talk a little bit about the length tension relationship and also the force velocity relationship of concentric and eccentric muscle contractions, and how to take advantage of the speed of training so that you can increase both the concentric and eccentric muscle contraction ability in the muscle fiber. All right, so let's move on here. And we're going to talk about this length tension relationship. So your length tension relationship. Now, here's another little interesting connection here. There's a connection between length and tension, or length and tension create this system, and what emerges from the system, like what emerges from Cassie and Daniel connecting, is this relationship. There's also an emergent relationship between length and tension. And also here's another throwback to my dynamic systems theory or systems theory, where there's another relationship that develops between speed and force. So let's just go take a quick look into those relationships here. So now we're only talking about an active process right here. And we're talking about the actin and myosin intertwining to create a force. But not only is there the active component of muscle, there's also the passive component. And we're going to have to talk about both of them. And I'll use this graph to explain what's going on. Anybody have any questions before we move on to this? Everybody's good? All right, I know there's a lot of stuff, but I'm recording the screen and I'll post today's and Monday's lectures so you can go through it a million times if you need to. All right, this is a relationship. This graph emerges. I don't think anybody's ever thought about this graph in their life, but this happens all the time. And what it does is it makes it so that you can move, or your movement patterns kind of are dictated by this relationship. And I'll explain what I mean. All right, so it says sarcomere length here. This means the length of your muscle. Now, when my arm here, this is my upper arm, this is my lower arm, this is my hand, all right? And I'm just gonna draw my bicep here, my bicep. Like this is my bicep and my bicep tendon. Now, this is about 90 degrees, 90 degrees of my arm flexion. I could also extend my arm, let's say, all the way. And if that's the case, I have my shoulder here, and this is my elbow, and this is my forearm, this is my hand. And then if that's the case, my bicep is going to be extended, stretched all the way. It's going to be nice and long. All right, it's going to be long. This is my bicep now, long. My arm is fully extended. Or I could also have my arm in this orientation here, where my arm is, this is my upper arm, my humerus, this is my forearm, this is my hand, whoopsies, and you understand. So this is, my arm is totally flexed. This is at the middle of my arm's length, like my arm's range of motion, and this is extended as far as possible. In this situation, I may as well finish the drawing. In this situation here, my bicep is going to be relatively short and compact. All right. And as I extend my arm further, it's going to elongate and elongate. So my arm being completely flexed is when my muscle is going to be as short as possible, when my sarcomere length is really short. All right. This situation here, and I'll mark this with an A. All right, now B here is this situation here. And that's when my muscle is at about 40 to 60% of its like my limb or my muscle is at 40 to 60% of its length. And we'll also go like this, C, and this is C here. So A, B, and C. And C is when my length, my muscle length is going to be kind of like in this situation here. And maybe I'll also say that C elongated is like this. So whoops. This is C. And this is A down here. And when my muscle is right in the middle of its length, I would say that it's kind of in this range here. All right. Now, what it says is, if I have to find where the ability to create force or tension in my muscle is highest, I would say it's probably right around this range right here. That's when the tension or the ability to develop force in my muscle is highest right here. If I trace that graph, I'll go like this. All right, that's where the tension is highest on this graph right here. The tension is lowest over here and over here. All right, and if I trace both of those over, it says I can produce zero tension. So zero tension. Now, what happens to this napkin here if I can't create any tension when I pull on it? What's going to happen? It's just going to rip because it can't develop any tension. So right here it says that the muscle cannot provide any tension. And again, the muscle cannot provide any tension over here. So why? Why can it produce a huge amount of tension in the middle of its length? Well, that's because in the middle of its length, most of the actin here can attach to most of the myosin. When my muscle is overextended, like my arm is almost completely extended, very little of the actin and myosin can intertwine with each other. There's a very small overlap here. And as my muscle contracts more and more and more, contracts more and more and more, gets smaller and smaller. As the sarcomere gets smaller, more and more of the actin and myosin can interact with each other. More and more and more. The circle gets bigger and bigger and bigger. So as my muscle goes from contracted and gets longer and longer and longer, right about to here. Actually, I'm gonna stop the screen share for a second because this right here is a little bit difficult to understand, this graph. So I'm gonna stop the screen share for a second. When my muscle is fully contracted all the way, let's say this is the actin and this is the myosin. Again, it's just a model, it's not real. This isn't what it really looks like. All right, right now the muscle is fully extended all the way. And the actin and myosin just can't reach each other. So if they can't reach each other, they can't pull and create a tension. When your muscle is halfway, a lot of the actin and myosin can overlap with each other so that they can pull on each other. But then when your muscle is completely contracted, what happens is the actin and myosin, they pass each other. And I still can't create an active contraction because the actin is over here and the myosin is over here, and they still can't contract. So either my muscle is too extended and I have no cross bridges to interact with, or my muscle is too contracted. And again, the actin and myosin have just totally slid past each other. So I can't create an active contraction. That's going to be this example here. All right, how do we do that again? That's going to be, oh, it erased it all. All right. that's going to be this example here. When your muscle is completely contracted, you have too much overlap. None of the actin can touch the myosin, even though that's not what this graph here shows you. All right, right here, I can't create a contraction because none of the actin and myosin are interacting. And if I look at stage 1 here, this is when my muscle is completely extended. And again, none of the actin and myosin are interacting. So I can't develop an active tension. So in the middle here, though, let's go. I just want to make sure that this is clear. We're only talking about the muscle's ability to actively create power. And it's active because you're using ATP. So right now, if I want to create the most amount of force, I'm going to try to do it with my muscle in this orientation here, 90 degrees. Because at 90 degrees, my bicep has the greatest overlap between my actin and myosin. And the further and further I extend my arm, the further and further my arm gets extended, or the further and further my arm gets contracted, the less actin and myosin cross bridges there are. Until finally, I cannot produce any force when my muscle is completely contracted, and I cannot produce any force when my muscle is completely extended. That's why everybody, take your arm and completely extend your arm like this, all the way. Just put it out in front of you and extend your arm all the way, and now try to tighten your bicep muscle. You can't do it. It's because none of the actin and myosin can touch each other. And if you're trying to make the biggest muscle, what you're going to do is not all the way like that. Actually hurts my elbow. You're going to do it sort of at 90 degrees. And when you're at 90 degrees, you can feel the contraction happening. It's because all the actin and myosin are overlaying with each other, and they can actually make a cross bridge. There can be recycling of the ATP, the actin and myosin can bind. So now, if I was going to say, throw a book bag at you, all right, and you've never ever, let's say Jenny, you and I walk into the, you walk into the classroom, and it's the very first day of class, and I'm holding my book like this, and I go, hey Jenny, catch, boom, and I throw the book, and you're like maybe like 3 feet away from me. You had no idea I was going to throw the book at you. It wasn't something that you could necessarily have this brain contemplate what's happening, send the action potential, and predict what I'm doing. How would you catch the book? Or Morgan, I took my computer. You know my computer is something that's like valuable to me. It has my entire lesson plan, all that stuff. I just, out of nowhere, take my computer and I throw it at you. Or Cassie, I take a heavy bag of rice and I throw it at you like this. How are you going to catch it, Cassie? Show me. You're going to catch it with your arms at 90 degrees. Why? Why would you hold your arms at 90 degrees to catch something? And where does this come from? Is this the brain saying, oh my God, 75 degrees, not good. 110 degrees, not good. Got to be 90 degrees. Why would it be 90 degrees? And where does 90 degrees come from? Especially, Cassie, when I threw that bag of rice at you, and it was so fast that you didn't have time to think about sending an action potential, think about getting your arms 90 degrees. And in fact, if I'm going to throw anything at you, you're really going to carry it at 90 degrees. Why? Is that a cognitive thing? Is that something that you're deciding to do? For me, it seems as though that 90 degrees has always just happened. It always happens. And if I don't catch it at 90 degrees, usually what's going to happen is it's going to either drop the object, or I'm going to hurt my arm. I'm going to hurt my bicep. Why? Where does the 90 degrees come from? Well, I'll tell you, the 90 degrees comes from here. It's an emergent motion. It's an emergent pattern that happens because if I don't catch it at 90 degrees, my muscles can't produce any tension. And if my muscles can't produce any tension, what happens is my muscle rips. I have no other option. I don't have an option to catch it at completely contracted. And I also don't have the option to catch it with my arms completely extended, because I don't have the ability to actively produce a contraction at that stage of the game. 1 second. 1 second. Silly me. All right, silly me. I have to screen share again. So you're not going to catch a weight with your arm fully extended, or a bag of rice with your arm fully extended, because you can't produce any active contraction. There is no active contraction here. You can't produce a force. And you're also not going to catch the weight with your arms at fully contracted, because again, you can't produce any force. So the fact that your muscles and your actin and myosin, your sliding filaments, your excitation coupling system functions this way, what emerges is this length tension relationship. And what emerges off of that is your posture or your moving strategies, right? That's why you never have to think about catching something at 90 degrees. That's why you've never contemplated catching something at 90 degrees. You catch it at 90 degrees because you have to. You catch it at 90 degrees because you have actin, myosin, because you have cross bridges and ATP. You catch it because you have bioenergetics, because you have a system here. And what happens is, what emerges from this system of physiology is the movement strategy at any given time that you need in order to really perform and do the things that you're doing. So this length tension relationship, the relationship between your orientation of your actin and myosin, the relationship between length and the ability to produce force, allows for your posture or your movement strategies to emerge. It's why you've never ever thought about it. And even from a little baby, that's the way that your body would work. To me, this is really amazing. All right, now, why isn't it the case? So let's talk about one last thing that's related to this. This is the active component, right? The active component, meaning this is what happens when your muscles actively produce a tension. Now, they actively produce a tension. Now, let's say, for instance, I go like this, all right? Maybe I won't stop the screen share. Can you guys see me from here? I'm sure you can. Just make sure you take a look. Now, it says that if my arm is fully extended, I cannot produce an active tension. And if I can't produce an active tension right here, the bicep, the tissue that is my bicep, the sarcomeres should absolutely just rupture in half, because I can't produce an active tension. But why is it the case that I can hold my book out like that then? My arm is fully extended. I can't produce an active tension in my bicep, but my bicep isn't exploding in half right now. Why? All right, because of the tighten in my muscles that's providing elastic energy. So when the body's ability to actively produce a tension kind of goes away because of the extension of my arm, remember, I can also produce a passive, let's pick a different color so it's clear. I can produce a passive tension. And that's why this tighten stuff within those actin and myosin filaments is so important. So I can produce a passive tension in my muscles. And I'm going to draw the passive graph. All right, the passive graph looks kind of like this. Slow, low, 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 low. And then as my muscle starts to elongate, the ability to store passive energy starts to increase and increase and increase. So take a look at the screen one more time so you can see what I'm talking about. This material has the ability to produce passive tension, just like your muscle can. Now, when the material is shortened as short as it can go, it's not producing any tension at all. There's zero tension in the muscle, zero tension in this particular material. All right, zero tension. There's slack in the material, actually, no tension though. That's when the material is as short as it can be, right here. But when I elongate the tension or elongate the material, you can see now I can create more tension. And the more I pull, the more I stretch, the more tension, until what's going to happen, Sydney? Eventually, what's going to happen?
It's gonna like tear. The sarcomeres are gonna separate.
Right, they're going to separate so much that there's going to be a rupture of this particular material. So right down here, when the muscle is contracted all the way, right down here, it cannot produce any of this passive stretch tension. But as the muscle gets longer and longer and longer and longer, like now, when the muscle is over here, when the muscle is in this orientation, it cannot produce an active tension, but the ability to passively stretch increases, increases, increases until there's no longer any active tension. It's just the material stretching and stretching and stretching and stretching. And eventually, if the material stretches so much because of an external force that's put on it, like if I'm doing a deadlift, and typically you're going to do a deadlift with your arms straight like this, one hand in a pronated grip, one hand in a supinated grip, but the force on the bar is pretty high, but my arm is extended all the way. So I go to lift the weight, and if the weight is too much and the passive ability for me to store energy in that muscle isn't enough, I'm just going to have a bicep rupture. I'm going to tear my arm. So right now your arm, and if I was to add these two graphs together, and I would get the total length tension curve from my muscle, it would look something like this. It would look like this. There is no contribution to the passive component here because it's zero. So all the way up until right about the number 2, it would only be active tension. And then as the active tension starts to drop down, you'll see the contribution of the passive graph starts to pick up, and the graph is going to start to go like this, 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 boom, until all of the contribution of tension, as my arm gets longer and longer and longer, all of the contribution of the tension that my muscle can provide is no longer active tension, it's only passive tension. And if I exceed this threshold of tension in my muscle and my tendon, what's going to happen here is a complete rupture. All right. So that's what the active length tension relationship of muscle looks like. And it comes because of this system between actin and myosin, ATP, recycling of energy. And what emerges from this is not length or slope. What emerges from this, I think, is super powerful. What emerges from this is the orientation in which my arm would assume in order to catch something and not overextend the passive component of that muscle. Oh, I think that's pretty powerful. All of a sudden, I started thinking of this stuff, and I stopped thinking that hierarchical motor programming was real. And I started thinking, wow, this is starting to add more stones into the systems theory bucket. Emergent postures, patterns that I don't really even think about. And Cassie, if I threw you that bag of rice and you had no time to interact or no time to think about it, you would still, boom, catch it like this. And if you didn't catch it like this, most likely you would drop it and you wouldn't be able to perform the task that you needed to perform. right? So this is an example of an emergent property of posture coming from this active length-tension relationship here. The system between length, or the constraint between length and tension. All right, so let's move on to the last slide of the day, and maybe next Monday I'll go through these stress strain curves, but I think that we've done a pretty good amount in terms of this stuff right now. So let's clear this up. Let's go to the last graph in the chapter, and we'll talk about that too. So this here is the force velocity relationship, the force velocity curve. All right, so let's just talk about, let's draw on this just a little bit to make it more clear. And let's draw a line down here. Now this line right here is force. So the higher up you go, the more force. And this is velocity. This is going to be positive velocity, and this is going to be negative velocity. Now velocity is equal to distance equals rate times time. So distance equals rate times time. And therefore, rate is equal to distance divided by time. And when I say a negative velocity, I mean a negative distance. And a negative distance is if I'm going backwards, and a positive distance is if I'm going forwards. In muscle contractions, negative distance is when your muscle is elongating. Negative equals eccentric. All right, because your muscle is moving in the eccentric or negative direction, and positive is equal to concentric. But if I say, for instance, drive my car 100 miles an hour forward, or I drive my car 100 miles an hour backwards, positive 100 miles an hour is the same thing as negative 100 miles an hour. It's just a different distance, a different direction. So let's take a look at this side right over here first. I wish I could do this. I bet I can. Let's take a look at this side first. Okay, let's take a look at this side first. This is the force velocity relationship for concentric muscle contractions. And it says, and this is an isometric force here. Whoops, let's go undo that. And let's go back to here. Let's go back to drawing. We'll use a different color. Right now it says that this is the amount of force that I can produce on an isometric contraction. And remember, an isometric contraction has zero velocity forward or backward. So an isometric contraction here is always more forceful than on this side, a concentric contraction. That's what it's saying. And a concentric contraction is always more forceful. Well, we'll see where force is highest on this graph. I start down here, it's lowest, 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 and it's highest right here. This is where the force is highest for a concentric muscle contraction here. And it says that a concentric muscle contraction is most forceful at very low velocities. That's why if you do a bicep curl and you have a lot of weight, you're not going to do the bicep curl very fast. It's going to be slow. Because the way that your muscles fire concentrically is that it can produce the most amount of concentric force at low velocities. The faster and faster you move your muscle in a concentric contraction, the less force you can produce. So when you do a bicep curl, I've never said, okay, man, I'm going to have to do this bicep curl at 2 or 2/10 of a mile an hour, a meter per second. I never have ever thought that. It just happens. It emerges. It emerges because of the weight that I'm using, because of the excitation coupling, because of the ATP recycling in my muscle. It emerges. It comes from nowhere. So if I want to do a concentric muscle contraction, I'm going to be more forceful at low velocities. So your body does it at low velocities. All right, if I take, let's go like this and erase this. If I look at the opposite side and I just cover this up, and I'm just looking at my eccentric muscle contractions, it looks like an eccentric muscle contraction is the opposite. An eccentric muscle contraction, first of all, is always more forceful than an isometric contraction. Oops. Draw this. Is always going to be more forceful than an isometric contraction. That's why I don't use isometric contractions or lock my legs out when I jump off a rock wall. And it says right here that the highest force I can produce in an eccentric contraction is actually at higher velocities, which is why when I jump off a rock wall, I don't try to absorb that landing slowly and go down into a deep knee bend, because I can't produce as much force. So if I can't produce as much force with my muscle, then what's going to happen is that force is going to be absorbed by my ligaments and my cartilage. And I don't want that. So an eccentric muscle contraction is always more forceful than its counterpart in the same muscle in the concentric contraction. And eccentric muscle contractions are always more forceful at higher velocities. All right, so let's erase this here. Now, if I'm a strength and conditioning coach, what I usually have people do is fast contractions concentric and slow eccentric contractions. Like you have somebody do like a pull-up and they do the concentric phase fast and they do the negative slow. That sounds a little bit counterintuitive, because my concentric contractions are better at going slow and my eccentric fast is better at going fast. So why wouldn't I do a bicep curl slow and the eccentric lowering part fast? Well, because your muscles are already able to produce higher forceful contractions slowly. So if I do a muscle contraction that is, whoopsies, redo that, undo that. If I do a muscle contraction in concentric that is faster, what's going to happen is you're going to start to increase this graph. And this graph is going to start to kind of look more like this. So in the gym, I'm going to have my athletes do fast concentric contractions and try to increase this area here. And in eccentric contractions, I'm going to try to get them to be a little bit slower. So now I'm kind of filling up this graph here. And now what you're doing is you have a strategy to improve or increase your overall strength of both concentric and eccentric muscle contractions. But now I'm not necessarily telling people to do fast concentric and slow eccentric because I want them to. I'm doing that because that's a teaching strategy that emerges from this force velocity relationship. It's a teaching behavior that I have to adopt so that I can improve the overall strength and conditioning of my athletes. Right? So now all of a sudden you're going to see your own teaching, your own work in physical therapy, Tatiana, as you get to grad school. A lot of the things that you're going to learn in this book alone are emergent properties or emergent teaching strategies that are dependent upon actin and myosin, not the brain doing it. It's the active ability to produce force, or the passive ability to produce tension, or the recycling of ATP that is more a function of why you would catch something this way or why you would teach something that way than it is your brain sending signals to your body to accomplish those motions. Right? So I think that that's probably enough for today, honestly. I think that we just went through a good amount of information. I think we got through all of chapter 1. Does anybody have any questions about anything, any content or anything like that? Drew, we Gucci, baby?
Yeah, absolutely.
All right, man. I hope you guys have a great weekend. Enjoy the time that you have with your family. Stay healthy, make good choices. All right. If you need me for any reason, email me or it's easy. Just send me a quick text message, whatever. Okay. So next week, what we'll do is I'd like to try to get into the stress strain relationships, because I think that they're really informative. And I think that they're helpful in terms of you understanding a little bit more about like tissue mechanics and things like that. So we'll start with that at the beginning of the week. Then we'll get into bioenergetics. Bioenergetics is carbohydrate, protein, and fat. And for the most part, the suggestions that I would give for exercise physiology and for sport performance are going to be totally different than the suggestions that you would give for the regular population of people. The regular population of people is 60 to 80% overweight and obese, and 50% of those people don't do any exercise. So for those individuals, what I would do is I would cut down on carbs, because carbs become fat, adipose fat. But for an athlete, with my guys spending 4,000 calories a day, I want those guys housing brown rice and quinoa as much as possible, because sugar, carbohydrate, is fuel, and protein is scaffolding for your muscles. You do not want to use your scaffolding for fuel. It's like if you go to like a barbecue and there's like a bonfire and you have all this wood and you keep throwing the wood into the fire. The wood is like carbohydrate. You keep throwing the wood in, but then when there's no more wood, Cassie, what do you throw in? The chairs, right? The cans that you have, anything else to keep the bonfire going. And your body is very similar. If you have no more fuel, your body's going to start burning its own fat stores, and then it's going to start burning its own protein, right? It's going to burn its own muscle. So you don't want that. Okay. All right. I appreciate the job very much. I hope you guys have a great weekend. I look forward to Monday. All right. Make good choices.
Thank you.
Okay. You're very
Thank
welcome.
you.
Take care.